Hello everyone. A very good evening to all of you. Am I clearly visible, audible? Let me confirm. If I'm clearly visible, audible, I will start the lecture ahead. Give me a minute to confirm it. Yes, I hope it is working. So I welcome you all for today's session. A very good evening to all of you. I am Dr. Priyanka Sachdev here, and today I'm here to continue my series that is super simplified pathology. So in this series, I'm going to cover entire Robbins in a super simplified manner on free YouTube lectures. So we have started general pathology. In pathology, we have three portions: general pathology, hematology, and systemic pathology. One by one, we will cover all in this series. So I have started general pathology. In general pathology, we have started neoplasia in the morning. So we have seen few topics from the neoplasia. So this is a lecture in which I'm going to teach you the molecular genetics of neoplasia, the most difficult portion of the entire general pathology, I must say. As per my knowledge, it is the most difficult portion. I will try my best to super simplify all the genes in front of you. Today in this lecture, I am going to teach you how mutations of various genes leads to various cancers. I will discuss many proto-oncogenes like RAS, like ABL-BCR, like MYC gene. I will discuss many tumor suppressor genes like retinoblastoma, RB gene, and I will discuss some DNA repair genes also like P53 genes and apoptosis regulating gene. So I'm sorry. I'm going to discuss all these genes one by one. So let me start neoplasia. As we have discussed in the morning that what is neoplasia. Let me define neoplasia. Neo means new, plasia means growth. So any new growth in the entire body is known as neoplasia. It is known as neoplasm. The new growth is known as tumor. The new growth can be benign, can be malignant. That is, tumors or neoplasm can be benign, can be malignant. In our last lecture in the afternoon, we have seen the differences between benign and malignant tumor, how to differentiate benign and malignant tumor microscopically. So it is the anaplasia. Anaplasia is the key to differentiate benign tumors. Just a second. Benign tumors from malignant tumors. So anaplasia is the hallmark of malignancy. Anaplasia is absent in benign tumor and present in malignant tumor. The branch of science which deals with neoplasia is oncology. We already know that. Now in the morning we have seen that all cells in human body, the normal cells, I'm talking about normal cell, not cancer cell. Now let me come on the genetics. No, all the normal cells in human body have growth receptors on their surface. Can you see the growth receptors on the surface? Can you see the growth receptors on the surface of a normal cell? Right. When growth factor is coming, that is stimulus is coming, binding with the growth receptor, at that point, this cell will do the mitosis. This cell is doing mitosis, is dividing into two cells. So this is a normal cell. This is a normal cell. But in neoplasm, what is the difference between neoplastic cell and normal cell? In the morning also, I have told you a neoplastic cell, see the difference. Even in absence of growth factor, that is absence of stimulus, the cell is doing the mitosis. So this is a neoplastic cell. It is not under physiological control. Even in absence of stimulus, it can divide. Now, how it is doing so? It is due to the mutation on the genes. So since this cell is doing uncontrolled mitosis, the mitosis is not under control. It is uncontrolled. It is uncoordinated. It is not under the effect of the stimulus. So whether stimulus is present or absent, it doesn't matter for the cell. The cell is dividing, dividing, dividing. From one cell, it is forming two cells. From two, it is forming four. For four, it is forming 16, 8, 16, 32 likewise. So a bunch of cells is formed, which is known as neoplasm. So what is neoplasm? Neoplasm is abnormal and uncoordinated growth of the cell, right? Growth of the cell. The growth persists even after cessation of the stimuli. So the stimulus is absent, even the growth is persisting, even after the cessation of the stimuli. That is. Now let me come on the genetic regulators or the genes, and that is molecular mechanism, how cancer occurs. There are total 4 genes. Now listen it like a story. I'm going to teach you a very interesting story today. No one will teach you neoplasia with such a beautiful story. Now listen the story. So there are 4 genes. There are proto-oncogenes, tumor suppressor genes also known as anti-oncogenes, apoptosis regulating genes, and DNA repair genes. These 4 genes, the mutation of these 4 genes will lead to cancers, will lead to neoplasia, will lead to tumors. Now, what are the normal function of all these genes? Let me explain. So, let me take 2 cells in front of you. Can you see 2 cells in front of you? Give me a thumbs up. Can you see in this diagram? You can see 2 cells. Both the cells are normal cells. I'm not talking about tumor cell. Both these cells are normal cells. Both these cells have growth receptor on their surface. You can see the growth receptor on their surface. Both these cells have growth receptor. Now, in the first cell, growth factor is coming. The growth factor is coming, binding with the growth receptor. So at that point, when growth factor is coming, binding with the growth receptor, this cell divide into two cells and mitosis takes place and it is dividing into two cells. So when growth factor is present, this cell is dividing into two cells. See the another cell, the second cell. Here no growth factor is present. So growth factor is absent. So absence of the growth factor, because of the absence of the growth factor, this cell is not undergoing mitosis. So how does cell came to know? How does this cell came to know that growth factor is arrived? Now I have to divide. I have to form two cells. I have to do the mitosis. And how does this cell came to know that the growth factor is absent and I don't have to divide? I don't have to do mitosis. So mitosis is absent. How cell came to know? Actually, the answer is genes. It is through genes. So let me come on genes. Can you see when growth factor is coming, when growth factor is present, inside the DNA of this gene, inside the DNA of this cell, there is a gene known as proto-oncogene which gets stimulated. And proto-oncogene is stimulating mitosis. Proto-oncogene is the gene which is stimulating the mitosis. It is causing the cell division and forming two cells out of this cell. So proto-oncogene is increasing the rate of the mitosis. Right? Now see the other way around. In the other cell, when no growth factor is present, when the growth factor is absent, another gene inside the DNA of this cell got stimulated. The name of the gene is tumor suppressor gene. So it is a tumor suppressor gene which is inhibiting mitosis. So tumor suppressor gene is a gene which decreases the rate of mitosis and proto-oncogene is a gene which increases the rate of mitosis. Till now the concept is clear to you? Till now give me a thumbs up. Manoj, the concept is clear to you? So proto-oncogene is a gene which increases the rate of mitosis. Tumor suppressor gene is a gene which decreases the rate of mitosis. Now listen the beautiful story. So can I say proto-oncogene is the gene which increases the rate of mitosis? So it is like the accelerator of a car. So accelerator increase the speed. So it is like increasing the rate of mitosis, increasing the speed of mitosis. And can I say tumor suppressor genes are the brakes of the car? Because when we apply brake, the speed is decreased. So whenever we drive any vehicle, I want to increase the speed, I will apply accelerator. So accelerator will increase the speed of the vehicle. And whenever I want to decrease the speed, I will apply brake. So brake will decrease the speed. So the speed is the balance. So my car is in balance. The speed is in the balance. Whenever I want to increase speed, I will apply accelerator. Whenever I want to decrease speed, I will apply brake and there is no accident. The speed is under balance, right? The speed is under balance. So proto-oncogenes are the accelerators of the cell and tumor suppressor genes are the brakes of the cell. Imagine a situation. You are sitting or someone is sitting in a car. In a car, the brake is failed. In such a car in which the brake is failed, the brake is already failed. The brake is not working. Brake is failed. You are applying brake, but it is not working. Or you are sitting in a car in which the accelerator of the car is stuck at the highest speed. You cannot decrease accelerator. It got stuck at the highest speed. So imagine one of the two situation or imagine both occurring together. You are inside a car and brake is not working. Number one, and accelerator got stuck at the highest speed. What will happen? A blast. Accident will happen. Uncoordinated, unregulated, excessive speed will be there. So car or the vehicle will be, here the car or vehicle is the cell. So inside the cell, two things are happening. So the two genes got mutated. So proto-oncogene got mutated and it become overstimulated. After mutation, the gene do not have normal function. So what is mutation? What is mutation? In mutation, the gene got mutated. In the mutation of the gene, either there is gain of function of the gene or there is loss of function of the gene. So normal function of the gene, no more. So after the mutation of the gene, either the function of the gene increases or the function of the gene decreases. So here, proto-oncogene mein gain of function mutation is taking place. So this proto-oncogene, it become overstimulated. Overstimulated, so it will do excessive mitosis. And tumor suppressor gene, loss of function mutation take place. So here loss of function mutation takes place inside tumor suppressor gene. So it become inactivated. That is, brakes are failed. They are not working and accelerator are overstimulated. They are stuck at the highest speed. So inside the cell, what will happen? Uncontrolled mitosis. They will increase mitosis drastically and they are supposed to decrease, but they are not working. So the mitosis will increase uncoordinately and a bunch of cells is formed, which is known as cancer. So basically cancer occurs either due to overstimulation of proto-oncogene. Can you see here? Either due to overstimulation of proto-oncogene, that is accelerators got stuck at the highest speed, or inhibition of tumor suppressor gene. that is brakes are failed, brakes are not working. So here gain of function mutation takes place in this gene, here loss of function mutation takes place in this gene. First give me a thumbs up if you got this point. So proto-oncogene में कौन सा mutation होगा? Gain of function and its normal function increases. ट्यूमर सप्रेसर जीन में कौन सा म्यूटेशन होगा? लॉस ऑफ फंक्शन म्यूटेशन टेक्स प्लेस इन ट्यूमर सप्रेसर जीन एंड इट्स नॉर्मल फंक्शन डिक्रीजेस. सो accelerators and brakes are non-functional now. One is over functioning, one is non-functioning. So it is taking place inside the cell leading to the cancer. Give me a thumbs up first. Give me a thumbs up. Everyone give me a thumbs up if you got this. So what you will do if it is happening in your car, what you will do? Will you drive that car? If you drive that car, you know the accident is going to happen. So speed will be uncontrolled. It is excessive and speed is not under control because accelerators are overworking and brakes are not working. So proto-oncogene is over-functional and tumor suppressor gene is non-functional. So what you will do? You will say, ma'am, I will take my car to the mechanic. I will not drive this car. So who is the mechanic of the cell? In the cell, inside the DNA, who is the mechanic? So here comes the role of the third gene. So the third gene in the cell, who is the mechanic of the cell, is DNA repair gene. So they will call the DNA repair genes. The DNA repair genes, that is P53. So this is the mechanic. This is the mechanic. They both will go to the mechanic. Please repair us. Please repair us. I am overstimulated. The proto-oncogene will say I am overstimulated and tumor suppressor genes say I am inactivated or understimulated. So please repair us. Please repair us. So mechanic, if the mechanic is live, the mechanic is functional, that will repair us. So P53 is the DNA repair gene, that is the mechanic of the cell. Imagine a situation, the mechanic is also dead. The mechanic, the mutation occurs in the mechanic. The mechanic is dead. Loss of function mutation takes place in the mechanic. So mechanics are not working. Mechanic is saying no. P53 is saying no. I am not functional. I cannot repair both of you. So what will happen? What you will do now? What is the fourth and the last option? So your accelerator is stuck at the highest speed, that is proto-oncogene may gain of function mutation leads to overstimulation of proto-oncogene. Your brakes are failed, that is tumor suppressor gene may loss of function mutation leading to inhibition of tumor suppressor gene. The mechanics are dead, that is DNA repair gene P53 is showing loss of function mutation and they are inactivated non-functional. Now the last option remaining, you will throw your car in the junk. You will keep your car in the garage and kill your car, destroy your car. So you want to destroy your car because there is no use of driving such car. You will have accident. You don't want the accident. So you want to destroy your car, but you are not allowed to destroy it and you are compulsorily, you have to drive it. So destroying means apoptosis. So the cell will try to do the apoptosis. The cell will try to do the suicide. Apoptosis means suicide. That is, that cell is trying to kill itself. So apoptosis regulating genes are also non-functional. So the killing is not allowed. The destruction of the car is not allowed. That is the fourth gene, the last gene, apoptosis regulating genes. These are the genes which are required for destroying the cell, but these are also non-functional. So cell cannot be destroyed. Now imagine a situation. The accelerator is stuck at the highest speed, the brakes are failed, the mechanic is dead and you cannot destroy your car. So there is no option, you have to drive it. And when you drive it, a blast is going to happen. So that is happening with the cell. Proto-oncogenes, they are over-functional. Tumor suppressor genes, they are inactive. DNA repair genes, they are inactive. Apoptosis regulating genes, they are inactive. So there are 4 types of genes. Have you got it? These are the names of the 4 types of genes. See in front of you. Everyone give me a thumbs up. So what are the 4 genes, you tell me? So it is like a simplified story. It is like a simplified story. Everyone give me a thumbs up. You got it? Yes or no? Say yes or no. So proto-oncogenes are the accelerators. Tumor suppressor genes are the brakes. DNA repair genes are the mechanics. And apoptosis regulating genes are the genes which cause destruction. They cause the destruction. So what mutation I am going to take place? We are going to do here. So proto-oncogenes gain of function mutation, so they become over-functional. Rest all loss of function mutation. So they are inactive. So proto-oncogene is overstimulated. Tumor suppressor gene, apoptosis regulating gene and DNA repair gene, they are inactivated. It will lead to cancer. Everyone give me a thumbs up. You got it? So that is the thing. So that is the genetic mechanism. So basically we can say the first 2 genes are more important. Proto-oncogene, these are the accelerators. These are overstimulated because of mutation. And tumor suppressor gene, these are the brakes. These are inhibited because of the mutation. So here gain of function mutation takes place. Here loss of function mutation takes place and it will lead to cancer. It will lead to malignancy. It will lead to cancer. It will lead to tumor. Give me a thumbs up. So that is the molecular mechanism. Now I am going to teach you two genes one by one. So first I will teach you proto-oncogenes and then I will teach you tumor suppressor genes. Give me a thumbs up. Should I start proto-oncogene? Let me start with first proto-oncogene. So overactivation of proto-oncogene will lead to cancer. But normally, do you have proto-oncogene? We all have proto-oncogene in our body, in all of our cells. From head to toe, we are made up of cells. In all of our cells, proto-oncogenes are present. And in all of our cells, tumor suppressor genes are also present, but they are not mutated. Once they got mutated, it will lead to cancer, right? So what is a proto-oncogene? The term proto-oncogene is given by a scientist. The name of the scientist is Harold Vermus and Michael Bishop. Please learn the name of the scientist. Ispe MCQ hai. Proto-oncogenes were discovered by number 1. So these are the normal genes which are required for cell proliferation. These are required for mitosis. So that means these are the accelerators. They increases the rate of cell division, but they work under physiological stimulus. What do you mean by physiological stimulus? When growth factor is coming, then only cell division takes place. Otherwise, cell division do not take place. So when stimulus is coming, they are doing replication, and without stimulus, they are silent. That is normal proto-oncogene. But when proto-oncogene got mutated, can you see proto-oncogene? If it got overstimulated due to mutation, it is known as oncogene. So can I say proto-oncogene get converted into oncogene because of mutation? So what is the difference between the two? Proto-oncogene works under physiological stimulus. So when stimulus is present, it will do the cell division. When stimulus is absent, it will not do the cell division. That is the division. The stimulation of the proto-oncogene is under physiological stimulus. But oncogene, it is not under physiological stimulus. It is not under physiological stimulus. So whether the stimulus is present or absent, it is doing cell division because the proto-oncogene is overstimulated, and overstimulated form is known as oncogene. So what all mutations takes place? There are 3 types of mutations. It can be point mutation, it can be amplification, and it can be translocation. These 3 types of mutation can lead the conversion of proto-oncogene and oncogene. Can I say proto-oncogene and oncogene both are same? Oncogene, proto-oncogene is present in normal human beings. It is present in you, me, everyone. But oncogene is present in cancer patient. So oncogene is nothing. It is the overstimulated form of proto-oncogene. So it occurs due to the mutation of proto-oncogene. Once the proto-oncogene undergo mutation, gain of function mutation, it will become overstimulated, and now it is known as oncogene, and it will lead to cancer. So in normal cell, it is known as proto-oncogene. In cancer cell, it is known as oncogene. Give me a thumbs up if you got it. Give me a thumbs up if you got it. 3 types of mutations are there, as I have told you. Point mutation, translocation, and amplification. So these 3 mutations, point mutation, amplification, and translocation, is leading the conversion of proto-oncogene into oncogene. Have you got it? Now come on the classification of proto-oncogene. There are 5 types of proto-oncogene. Let me divide the proto-oncogene into 5 categories. So these are the 5 categories, 1, 2, 3, 4, 5. You can see. Let me tell you the classification of proto-oncogene. Now how you will divide proto-oncogene into 5 categories. Listen, how a cell division take place. Let me draw a cell. This is a cell. This is the nucleus of the cell. Inside the nucleus, this is the double-stranded DNA. This cell want to do the cell division. How does it will occur? So initially, all cells have growth receptors on their surface, right? And initially a growth factor will come. Have you got it? So initially a growth factor will come. This growth factor will come and bind with the growth receptor. When growth factor will come, bind with the growth receptor, this cell will get the signal that I have to divide. I have to do the mitosis. I have to divide. This cell will get the signal that yes, I have to do the cell division. So what does the cell will do? The growth receptor, listen my words, the growth receptor is present on the cell surface, right? But the cell division, you have to give the signal to the nucleus that growth factor is already arrived. Now do the cell division. Do the replication of the DNA. Double the DNA. We have to do the cell division because growth factor is already arrived. So who will carry the signal? From the cell membrane to the nucleus, there are certain proteins, a chain of proteins which will carry the signal from the cell membrane to the nucleus. That growth factor is already arrived and now we have to do the mitosis. This cytoplasmic proteins which are carrying the signal from the cell membrane into the nucleus, these are known as cytoplasmic transduction protein. Cytoplasmic transduction protein. Have you got it? These are cytoplasmic transduction. So first step is growth factor arrival, number 1. The second step is binding of the growth factor with the growth receptor. The third step, the cytoplasmic transduction protein got activated and they will carry the signal from the cell membrane to the nucleus. What is the signal? What signal they are carrying? That growth factor is already arrived. Now start cell division. So nucleus will receive the signal. On receiving the signal, nucleus will do two things. What two things nucleus will do? Nucleus will do transcript. Now genes inside the nucleus will got activated. Which genes I am talking? So inside the nucleus, the proto-oncogene will get activated, which causes cell division. So all genes expressed by doing transcription and translation, they form protein. So proto-oncogene will also get activated with the help of transcription. The mRNA will be formed and mRNA will come out in the ribosome and protein will be formed. So transcription and translation. So the next step is transcription and translation. With the help of transcription and translation, protein will be formed. More and more protein will be formed. And which protein will be formed? In the end, the name of this protein, these are CDKs. These are CDK protein which will do the cell, cyclins and CDK, and they will do the cell division. They will do the cell division. Everyone give me a thumbs up. So there are 5 steps. If this cell want to divide, the 5 steps have to be followed. Can anyone name the 5 steps? What are the 5 steps? Tell me. So initially, a growth factor is coming. The growth factor is coming and binding with the growth receptor. The second step. The growth receptor, when growth receptor combination takes place in the cytoplasm, certain proteins got activated. The name of this protein is cytoplasmic transduction protein. They carry the signal from the cell membrane to the nucleus that the combination is already there, growth factor has already arrived, now do the cell division. So that is the third step in which there is involvement of cytoplasmic transduction protein. Right. The fourth step, there is stimulation of proto-oncogene. This proto-oncogene express itself with the help of transcription and translation. So fourth step is transcription and translation takes place. You know transcription. Transcription is formation of mRNA from DNA. Translation is formation of protein from mRNA. Ultimately, proteins are formed. Which proteins? Name them. The fifth and the last step, CDKs and cyclin. These are cell cycle regulating proteins are formed. And ultimately, this cell will divide into 2 cells. So cell division takes place. Give me a thumbs up. This all is happening due to proto-oncogenes. So there are 5 steps. So there are 5 type of proto-oncogenes. So proto-oncogene, as I have told you, listen again. As I have told you, listen the same story again. So this is the story. So as I have told you previously also, proto-oncogenes are the accelerator of the car. They increases the rate of mitosis. They increases the rate of cell division. Like accelerator of the car increases the speed of the car. In the same way, the car here is the cell. The cell is the car. So inside which we have accelerator, inside car we have accelerator. So inside cell we have proto-oncogene. Proto-oncogene increases the speed of mitosis. Like car increasing the speed of the car. Right? And like accelerator increasing the speed of the car. And tumor suppressor genes are like brakes. They are like brakes. So in the car, brake decreases the speed of the car. So tumor suppressor gene decreases the rate of mitosis. They inhibit mitosis, they decrease. Now, as I'm saying, imagine a car in which the brake is failed. Brake is non-functional. It is failed. And accelerator is stuck at the highest speed. You cannot decrease accelerator. You cannot. It is stuck. It got stuck at the highest speed. So in that car, uncontrolled speed is there. You cannot apply brake and you cannot decrease accelerator. What will happen? Uncontrolled speed, uncontrolled mitosis, uncontrolled. So if it is happening in a cell, the proto-oncogene become overstimulated and the tumor suppressor gene, they got understimulated or inhibited. It will lead to uncontrolled mitosis that will lead to cancer. So basically, I am teaching you proto-oncogene right now. So in proto-oncogene, you have to do overstimulation of proto-oncogene due to mutation. So overstimulation is due to a mutation. There are 2 types of mutations in human body, gain of function mutation and loss of function. Mutation occurs at genetic level, genes. So if any gene is there in human body, any gene is there, the gene is performing its normal function. This is the normal function of the gene. But if the gene got mutated, so 2 options are there. Either the mutation is gain of function or the mutation is loss of function. Gain of function, after mutation, the mutated gene will do the same function but in excessive manner. Now the function is excessive, that is overstimulated gene is there. And loss of function, the same gene will do its function, the same function, but less or no function. So gain of function, loss of function. So I am saying that in proto-oncogene, gain of function mutation takes place. That is, they become overstimulated. That is, the accelerator got stuck at the highest speed. In tumor suppressor gene, loss of function mutation takes place. That is, these are the brakes. These are the brakes and they are failed. So here inhibition of tumor suppressor gene takes place after the mutation. Give me a thumbs up. So I am teaching you the various types of proto-oncogene. There are 5 categories in proto-oncogene which I have told you. This is the classification. You have to learn the classification. Now proto-oncogene, let me tell you the term proto-oncogene is not one gene. It is all these genes. It is a cluster of genes. all the genes which increases the rate of mitosis, it is a proto-oncogene. It is not a one gene. Remember this, learn this, understand this. It is an umbrella term containing many genes. So how many genes are there? What is the classification of proto-oncogene? What is the classification? So the 5 steps I have told you. In the cell division, there are 5 steps. So the first thing, you should have a growth factor. So can you see a growth factor here? Growth factor should come. A growth factor should come. Right. The second step is the growth receptor. It will bind with the growth receptor. Can you see the yellow color growth receptor is there? It is the growth factor is binding with the growth receptor. A combination is there. So first a growth factor is there. Second, a growth receptor is there. They are combining with each other. So the third thing, in the cytoplasm, certain proteins are there that will carry the signal from the cell membrane into the nucleus. They are carrying the signal that growth factor is already arrived. Now do the cell division. So the third step is cytoplasmic transduction protein. Cytoplasmic transduction protein, as I have told you. The fourth step, the genes in the nucleus will get stimulated. The genes will do transcription. They will do transcription and translation, and they will form proteins. In the end, proteins are formed. That is cyclins and CDKs. Give me a thumbs up. What are the 5 steps? So the 5 steps, as I have told you previously also, this is a cell. It want to do the cell division. So it occurs in 5 steps. First, a growth factor will come. Can you see this is a growth factor? This is the first step. This is the second step. It will bind with the growth receptor. It will come and bind with the growth receptor. The second step, because of this combination in the cytoplasm, we have various proteins. The name of these proteins is cytoplasmic transduction protein. They will get stimulated. What is their job? They carry the signal from the cell membrane to the nucleus. So these are cytoplasmic transduction proteins. Now the fourth step, the signal is received by the nucleus. Nucleus got the signal. Now on getting signal, certain genes inside the nucleus will get activated, and these genes will do transcription. So the fourth step is transcription and translation of the genes. That is the fourth step. And due to transcription and translation, the last step, the proteins are formed. The proteins. So it is cell cycle regulating proteins. So see, the 5 steps are in front of you. Growth factor, growth receptor, cytoplasmic transduction protein, transcription, the genes involved in transcription, and the cell cycle regulating proteins. Everyone give me a thumbs up. I tried hard here. Come on. So there are many genes under each category. This is the name of the category. Now give me a thumbs up. Am I visible, audible? Is anyone listening to me? Let me see if I can see your chat. Yes, I can see your chat. Give me a thumbs up. Come on. So have you got it? So let me tell you the names. So growth factor are of 4 types. Growth factor, the first category, PDGF, platelet derived growth factor, TGF, tumor growth factor alpha, right? FGF, fibroblast growth factor, and CMAT. So please learn PDGF, TGF, FGF, and CMAT. These are the 4 growth factors. You have to learn. It is rectification, right? But you have to learn the classification. Growth receptors are EGF, c-KIT, RET, and FMS, right? Cytoplasmic signal transduction are most important one. The 2 most important. One is RAS, one is ABL-BCR. I will give you the description of these 2, right? Transcription is MYC. The 3 type of MYC, c-MYC and MYC-L-MYC. And cell cycle regulating proteins are CDKs and cyclins. Cyclin D, cyclin A, and CDKs. Give me a thumbs up. So that is the classification. You have to learn the classification. You cannot escape it. Now in this classification, I cannot teach you all the genes. I will teach you only important one. So what is important at MBBS level? What you should know? You should know. So you should know 3 among them. The RAS, the most important one. ABL-BCR, the second most important one. And MYC, the third most important one. So I am going to teach you 3 genes. The RAS, ABL-BCR, and c-MYC one by one in proto-oncogene. The RAS gene, ABL-BCR gene, and MYC gene. I am going to teach you these 3 genes one by one. Give me a thumbs up. Give me a thumbs up. Let me start with RAS gene. The first gene is the RAS gene. Let me start with RAS gene. So let me start with the first gene, that is RAS gene. It is a signal transduction, cytoplasmic transduction protein gene, right? It is the most common proto-oncogene which get mutated. Among proto-oncogenes, among all proto-oncogenes, the most common proto-oncogene to get mutated is RAS. It is an MCQ. It is a previous year question. Most common proto-oncogene कौन सा mutated होता है? I will tell you the name of most common tumor suppressor gene also and overall also. I will tell you 3 questions. Most common proto-oncogene to be mutated in human? The answer is RAS. Most common tumor suppressor gene to be mutated in human? The answer is P53. And overall gene most common mutated in human? So answer is P53 again. So have you got the 3 answers? So RAS is the most common proto-oncogene to be mutated in humans. It is a previous year question. Learn it. right? Let me tell you two scenarios now. Now it is the super complicated, very complicated topic which is given in Robbins. But after watching my lecture, please read today only this topic from Robbins, please. So I will teach you two scenarios now. What happens normally inside me, you, we all have RAS, na? We all have RAS. So we all have RAS inside us, how it is functional, how it is doing the cell division, right? But once it got mutated, it can lead to cancer. So during mutation, what happening so that it is leading to cancer? So I will teach you two scenarios. Now let me start with the first scenario normally. So consider this scenario taking place inside a normal human being, inside you, inside me, inside all of us who don't have cancer, right? So normally, can you see a cell? Now this is the sketch diagram I have drawn for you to simplify the concept for you. So these diagrams are nowhere given. I have drawn super simplified diagrams for you to explain you the concepts. Can you see a cell? Yes, of course you can see a cell. On the surface of the cell, can you see a growth receptor? Yes, the growth receptor is present. On the inner side of the cell membrane, can you see a protein? It is RAS protein. It is inactive right now. Why it is inactive right now? It is bind with GDP. Now there is a rule. When RAS protein is bind with guanine diphosphate, it is inactive. It is inactive. And when RAS protein, RAS gene or RAS protein bind with GTP, it is active. It is active. What is GDP and GTP? GDP is guanine diphosphate. GTP is guanine triphosphate. So when two phosphate groups are present, it is inactive. When three phosphate groups are present, it is active. Have you got it? Count the number of phosphate groups here. There are two phosphate. That's why this RAS is inactive. Give me a thumbs up. Till now, is it okay? Now, in the next step, a growth factor is coming. Can you see a growth factor? Yes. A growth factor is arrived. When the growth factor is coming, this growth factor is binding with the growth receptor. So, GDP is replaced by GTP. Can you see the two phosphates are replaced by three phosphates? So, inactive RAS become active now. So, RAS is becoming active now. Give me a thumbs up. This active RAS will come in the cytoplasm. This active RAS will come in the cytoplasm from the cell membrane. Can you see this is coming in the cytoplasm? Now, after coming in the cytoplasm via various mechanism, don't learn the name of pathways. There are various pathways, RAF, MAPK, PI3K. These are all molecular level pathways. Via all these pathways, ultimately RAS is doing mitosis. So this cell, you can see a cell now, it will divide into two cells. Because of the RAS, it will divide into two cells. It will divide into the two cell. Now listen the story ahead. You will say, ma'am, cell division is already over. Now what ahead? Iske aage kya? You cannot keep the RAS. No, this is activated RAS. Now you cannot keep it active forever. You cannot active. You have activated it when growth factor is coming. When no growth factor is there, it was inactive. So again, after cell division, you have to inactivate it. My point is that one growth factor causes one mitosis. Causes now the one mitosis is over. One mitosis is over. So immediately after mitosis, one thing will happen. What will happen? Inside the RAS, there is a protein. The name of the protein is GAP, GTPase activating protein. What is the name of the protein? It is GTPase activating protein. It is GTPase activating protein. What does it mean? GTPase. It will split GTP into GDP. This protein will convert GTP into GDP. So immediately after one mitosis, GAP will come in role, and GAP convert GTP into GDP. So active RAS again become inactive. So this is the story now. Now the cell is already divided. So if this cell want to do another cell division, another growth factor, you have to bring another growth factor. One growth factor causes one mitosis. If you want another mitosis, you have to bring another growth factor, because RAS is under physiological control. So growth factor causes the cell division, and it is happening in me, you, everyone. One growth factor causes one mitosis. Give me a thumbs up. Everyone give me a thumbs up. This is normally. So the same diagram is given in Robbins. So this is how you have to write it. So when the RAS is inactive, the RAS is bound with GDP. Inactive RAS, the RAS is bound with GDP. Whenever growth factor is coming, at that time, GDP is replaced by GTP. So inactive RAS become active now. Now the RAS is active. Active RAS will come in the cytoplasm, and via various pathways, that is RAF, PI3K, there are various pathways, MAFK. So via these pathways, mitosis will happen. So one growth factor leads to one mitosis. Immediately after mitosis, there is an intrinsic protein inside the RAS which will get activated. The name of the protein is GTPase activating protein, right? This protein will degrade the GTP into GDP. So active RAS again become inactive. So that is the whole story which takes place normally. Have you got it? Give me a thumbs up if you got it. Anyone? Have you got it? So this is the normal story. Now let me come on mutation. What is happening in mutation? How it leads to cancer? How it leads to cancer? During mutation, now see the same diagram again and you will tell me where is the change coming. So see, again, this is a cell. You will say, ma'am, this is the same cell. This is the same cell. It is having a growth receptor on its surface. And the RAS is inactive because it is binding with two GDP molecules, two phosphate groups. Now growth factor is coming. You can see this is the growth factor. When growth factor is coming, GDP is replaced by GTP. Inactive RAS got converted into active RAS. This active RAS is coming in the cytoplasm and via various mechanisms, it is causing mitosis. Till now, everything is same. Now the RAS is mutated. What was happening next normally? After one mitosis, what was happening? I'm asking to you. After one mitosis, what was happening normally? In normal scenario, when the RAS is not mutated, what was happening? Please, anyone tell me? If you got it, anyone tell me what was happening normally? Normally, after one mitosis, the gap inside the RAS got activated. Gap. But here RAS is mutated now. So gap is mutated. Gap is non-functional. Gap will not come in the role. So if gap will not come in the role, GTPase will not come in the role. If GTPase will not come in the role, GTP will not convert into GDP and GTP will remain as GTP only. So if GTP will remain as GTP only, the RAS is active permanently. Now this RAS become active permanently. It is not becoming inactive. Yes, Zed. It is not becoming inactive. It is permanently active. So it will do uncontrolled mitosis. It will do mitosis, uncontrolled mitosis that will lead to cancer. So that is the thing. So when mutation is taking place in the RAS, basically due to mutation, the gap is non-functional. Give me a thumbs up. Zed, have you got it? I don't know your name. So have you got it? The gap is non-functional. If the gap is non-functional, GTPase will not act. If the GTPase activity is not there, GTP will not be broken down. It will remain as GTP only. GTP to GDP conversion do not take place. So there is permanent activation of RAS. GTP causes permanent activation of RAS. The permanently activated RAS will do uncontrolled mitosis and that will lead to cancer. I want a thumbs up. I want a thumbs up. Have you got it? That is the mechanism how RAS leads to cancer. So I have told you two scenarios. Normally when growth factor is coming, so RAS become active. After doing one mitosis, it automatically become inactive. That is activation of RAS in normally is transient. The activation of RAS is transient. Transiently, for a fraction of moment, for 1 second it is activated. After doing one mitosis, it again become inactive. That happens normally. That happens normally inside me, you, everyone. But during mutation, if the RAS is mutated, this is point mutation. So when one growth factor is coming, RAS will become active here also. But so it will do one mitosis. But after one mitosis, it will not become inactive because here gap is absent. Gap is mutated. So it will not become inactive and it will become active permanently. The permanently active RAS will do uncontrolled multiple mitosis that will result in cancer. That is the summary. Give me a thumbs up if you got it. Now tell me the examples. In human body, what cancers take place because of RAS mutation? There are three types of RAS: KRAS, NRAS and HRAS. There are three types of RAS genes. If KRAS is mutated, it will lead to colon, lung and pancreatic cancer. If NRAS is mutated, it will lead to melanoma and AML. And if HRAS is mutated, it will lead to bladder and kidney cancer. Everyone give me a thumbs up. I am done with RAS. I have given you the details of the RAS. RAS is done. So I have told you two things in the RAS. What two things in the RAS? In the RAS, I have told you what happens normally, what happens during mutation, and tell me the examples of RAS mutation, the examples of the cancer of the RAS mutation. What happens normally? You already know the story. What happens during mutation? The gap become non-functional. So the RAS become activated permanently. Permanent activation of RAS is taking place. And what are the examples? There are three types of the RAS: KRAS, NRAS and HRAS, mutation of which leads to tumor of various organs. Everyone give me a thumbs up. I really tried hard and I want you to appreciate my efforts. So I am done with the RAS. Should I come to the next proto-oncogene? After RAS, I would like to explain you ABL-BCR, the next proto-oncogene. Give me a thumbs up. Let me come on ABL-BCR. Let me come on ABL-BCR. Give me a minute. Give me 1 minute. Have you got it till now? Let me start. Give me a thumbs up. Come on. If you got it, what is the wrong in giving thumbs up? How I will know? I cannot see your faces now. I can see your chat only. You can see me, but I cannot see you. So how I will come to know that you got my point? You don't have any doubt. If you have any doubt, please write in the chat box. If you don't have any doubt, please give me a thumbs up so that I can go ahead. Give me the clarification. Okay, got it. So I want clarification from everyone. Listen, the next story, ABL BCR. It is very interesting story. Okay. I would like to draw a human cell. This is the human cell, any cell of human. All human cells are diploid. Yes. So this is the nucleus of human cell. What is the number of chromosomes? How many chromosomes are present in human cell? Can anyone tell me? How many chromosomes are present in human cell? I'm asking the number. How many chromosomes? Yes. So humans have 46 chromosomes in all cells. It is a diploid cell. Or you can say 23 pairs. You can say 23 pairs, right? 23 pairs. So just for suppose it is pair number 1. This is pair number 2, pair number 3, pair number 4, pair number 5, pair number 6, 7, 8, 9. Likewise, we have 23 pairs. I'm not drawing all 23, but yeah, we have 23 pairs. I'm not interested in all pairs. I'm not interested in pair number 1, 2, 3, all. I'm interested in 2 pairs. What are the 2 pairs I'm interested? I'm interested in pair number 9, and pair number 22. Pair number 22. Inside the nucleus, these 2 pairs I am interested. Learn the number. Chromosome number 9, the 2 pairs, the 2 chromosome in a pair, and chromosome number 22, the 2 chromosome in a pair. Normally, inside me, inside you, inside everyone, inside all normal human beings, we have a gene on chromosome 9. Just a second. We have a gene on chromosome 9. The name of the gene is ABL. It is present inside all my cells, your cells. ABL. And on 22, we have another gene. The another gene is BCR. This another gene is BCR. On chromosome 9, we have ABL. On chromosome 22, we have BCR. So they are performing their individual function. They are performing their normal function. These both are normal genes. Right. Now this is a cell. Let me draw the nucleus. They are present inside the nucleus. They are present inside the cell nucleus. Now mutation takes place. This cell is mutated. This cell is mutated due to physical, chemical or biological carcinogen. This cell is mutated. Due to mutation, now translocation will take place. Translocation. What do you mean by translocation? Now here, what's happening? Just a second. Here translocation will take place. Here translocation will take place. It is not functional. Just a second. Now here translocation. In translocation, a gene of chromosome 9 will go to 22. In exchange of it, a part of 22 will come on the 9. So in this translocation, ABL, the gene which is normally present on 9, it will go on 22. It will go on 22, and here after going on 22, it will bind there. On 22, we already have BCR. So a new gene is formed because of the hybrid of these two genes. Because of the fusion of these two genes, it is known as ABL-BCR fusion gene. Now this gene is deadly. It is abnormal. In exchange of this, in exchange of ABL, the chromosome 22 is giving an empty portion or non-useful portion to 9. So that is non-useful. So on chromosome number 22, a fusion gene is formed. A hybrid gene is formed. That is ABL-BCR. So that's why chromosome number 22 is known as Philadelphia chromosome. It is very important MCQ. Philadelphia chromosome. Please learn it. Have you got it? Have you got it? So because of formation of this fusion or hybrid gene, abnormal enzyme is formed. The name of the abnormal enzyme is tyrosine kinase, and that will cause uncontrolled mitosis that will lead to cancer. Everyone give me a thumbs up. So let me tell you 2 stories. What happens normally and what happens in mutation. What happens normally? What happens in mutation? Normally it's very easy. So normally we have 2 genes. What are the 2 genes? ABL gene present on chromosome 9 and BCR gene present on chromosome 22. They are doing their own function and it is normal. It will not lead to cancer. So you can see the diagram given by the author. The diagram given by the author. See normally. See normally. Don't see the mutation. See normally. Normally what is happening? So these are 2 chromosome 9 and these are 2 chromosome 22. This is 9. This is 22. On 9, if you can appreciate, please appreciate ABL gene. It is ABL. On 22, please appreciate BCR. This is normal. It is performing its own function. It is performing its own function, and mutation is under, mitosis is under control. Mitosis is under control. Now the mutation is taking place. Now mutation is taking place. In the mutation, you can see the ABL. ABL from here is going to 22. So on 22, please appreciate it is the combination formation. ABL BCR, the yellow blue combination. It is BCR ABL fusion gene, which is abnormal, and it will lead to abnormal tyrosine formation, tyrosine kinase formation. It will lead to uncontrolled mitosis and it will lead to cancer. So this is in mutation. Give me a thumbs up. So in translocation or in mutation, what is happening? So ABL gene, which is normally present on chromosome 9, it is translocated to chromosome number 22. In chromosome number 22, already we have BCR gene. So ABL BCR hybrid gene is formed on 22. It is an MCQ, not on 9. And chromosome number 22 is known as Philadelphia chromosome. Chromosome 9 is not Philadelphia because the fusion is formed on chromosome 22. In exchange of it, chromosome 22 is giving a non-functional, non-useful gene to 9. Right? So वो important नहीं है. The hybrid gene will form an abnormal signal transduction protein or signal transduction enzyme. That the name of the enzyme is tyrosine kinase. That is not written here. It will lead to uncontrolled mitosis. It will lead to cancer. Now these are the super simplified flow charts and diagrams. I am telling you, nowhere it is given. Please everyone give me a thumbs up. Please everyone give me a thumbs up. Everyone. What is the best example? If only one example, because of this hybrid gene formation, the person will have a blood cancer. There are 4 types of leukemia: AML, ALL, CLL and CML. So CML will happen. So please learn only one example. If it got mutated, the person will have CML. So ABL BCR translocation leads to CML. Only one cancer. In RAS, I have told you many cancer. KRAS के अलग हैं, NRAS के अलग हैं, HRAS के अलग हैं. There are many cancers. But ABL BCR mutation leads only to CML. Everyone give me a thumbs up. What is the treatment? You have to learn treatment also. There is a targeted therapy. The name of the therapy is imatinib. Imatinib is the name of the drug. It is the targeted therapy. It is the name of the drug, or it is the targeted therapy. What this drug will do? What this drug, the imatinib, will do what? The imatinib will inhibit the fusion gene. It will inhibit the fusion gene. So the targeted therapy is imatinib, and cure rate is very good. It is nearly 99%. It is the first targeted therapy which was discovered. इस पे भी बहुत PYQ है. Imatinib. That is the treatment of CML that inhibit ABL BCR fusion gene. I want thumbs up from everyone. So till now I am done with RAS gene. I am done with ABL BCR gene. In RAS also, in ABL BCR also, I taught you what happens normally, what happens during mutation, and the examples. Normally क्या होता है? In mutation, how does uncontrolled mitosis takes place, and what are the examples of RAS, the cancers which are caused by RAS, and what is the example of ABL BCR? Only one cancer, that is imatinib. So that is a summary till now. Let me come on the next. Till now we have covered 2 proto-oncogenes. Let me come on the next proto-oncogene. We have covered RAS. We have covered ABL BCR. Now I'm coming on the third gene, third proto-oncogene. The name is MYC. It is a MYC gene. Give me a thumbs up now. It's time to give thumbs up. Should I start MYC gene? Are you people there? Are you interested in the MYC gene? Let me start the third gene. That is the MYC gene. MYC. So let me start the third gene. That is the MYC gene. MYC gene is also a proto-oncogene. The MYC gene is also a proto-oncogene, right? It is also a proto-oncogene. So let me tell you 2 scenarios again. Normally, it is the same story now. Now take the 2 another chromosome number 8 and 14. Take the pairs. Again, humans have diploid cell. Inside the diploid cell, we have 23 pairs of chromosome. So this is pair number 1, pair number 2, pair number 3. This time, I am not interested in 9 and 22. I am interested in 8 and 14. I am interested in 8 and 14. Let me draw. This is chromosome number 8. The pair is chromosome number 8. And this is chromosome number pair 14. So the same story. Instead of 9 and 22, I am saying 8 and 14. Learn the numbers. Learn the numbers, right? On 9, there is on 8, there is a gene. The name of the gene is MYC. This is a normal gene present in me, you, everyone. On 14, there is another gene. The name of the gene is immunoglobulin. Immunoglobulin IgG gene, right? So that is on 14. So both of them are performing their normal function. So it is present in me, you, everyone. They are performing their normal function. But during mutation, if the mutation takes place, it is inside the nucleus. If mutation takes place, so it will lead to translocation. So again, a part of 8 will go to 14. In exchange of it, another part of 14 will come to 8. So that is balanced translocation. That is balanced translocation. So you all can see here MYC gene. The MYC gene is going from 8 to 14. The MYC gene is going from 8 to 14. Give me a thumbs up. So after going on 14, the MYC gene is fusing there. So already immunoglobulin gene is present there. So a fusion gene or a hybrid gene is formed on chromosome number 14, like it was forming on 22 in ABL BCR. Here MYC immunoglobulin hybrid or fusion gene is formed. That is abnormal. That can lead to uncontrolled mitosis, and that will lead to cancer. In exchange of this, the 14 is giving a non-functional portion to 8. So the same story. Give me a thumbs up. Only change is the number of chromosome. Instead of 9 and 22, it is 8 and 14, and name of gene is changed. Instead of ABL and BCR, we are having MYC and immunoglobulin. Give me a thumbs up. I cannot simplify than this. So normally, let me teach you normally. Normally on chromosome number 8, we have MYC gene. On chromosome number 14, we have immunoglobulin gene. And they are performing their own function. You can see the diagram here. You can see this is chromosome number 8. Both chromosomes of a pair is not shown. One chromosome is shown. So chromosome number 8 pe MYC gene is present. Chromosome number 14 pe immunoglobulin gene is present. So this is normal. This is all normal. Now see during translocation what is happening. During translocation what is happening. So can you see this MYC gene present on 8 is going to 14, and on 14 the fusion is formed. This fusion is leading to uncontrolled mitosis and it can lead to cancer. So in translocation what is happening? MYC gene which is normally present on 8, it is translocated to 14, and on 14 already immunoglobulin is present. So MYC and immunoglobulin hybrid is formed. This hybrid will lead to uncontrolled mitosis and it can lead to cancer. Give me a thumbs up, you got it. So till now we got 3. We got RAS, we got ABL-BCR, and we got the MYC. Now tell me the examples of MYC. MYC is of 3 types. Like RAS, it is also of 3 types. It is C-MYC, N-MYC, and L-MYC. Tell me the cancers from them. Tell me the cancers. L for L. L-MYC leads to lung cancer. N for N-MYC leads to neuroblastoma. And C is for B. C is not for C. C is for B. C-MYC leads to Burkitt. Right? L for lung. लंग में भी कौन सा लंग? इट इज स्मॉल सेल कार्सिनोमा ऑफ द लंग. ये तीनों पे एमसीक्यू है, सुपर डुपर इंपॉर्टेंट. ये तीनों पे पीवाईक्यू है, यू हैव टू लर्न. ओके, लेट मी टेल यू द समरी नाउ. सो प्रोटो ऑन्कोजीन इज ओवर फ्रॉम माय साइड. आई हैव टॉट यू proto oncogene completely. So I am done with proto oncogene. Let me summarize proto oncogene. You have to help me in the summary. Let me summarize. Have you got it? What is proto oncogene? Now I will start tumor suppressor gene. Have you got it? What is proto oncogene? Everyone give me a thumbs up. I will start tumor suppressor gene now. So proto oncogene is over. Okay, just a second. Okay. Let me see your chat. Yeah. So what is proto oncogene? Proto oncogene is like accelerator of the car. Proto oncogenes are the genes which increases the rate of mitosis, and they work under physiological control, right? So that is proto oncogene. Proto oncogene. Once they got mutated, here mutation is gain of function mutation. So they become overstimulated because of the mutation. After becoming overstimulation, they convert it into oncogenes, and these oncogenes will lead to cancer. That is the definition. We are done with the definition of proto-oncogene. After definition, we have seen the classification. The classification is front of you, and we have divided the proto-oncogene into 5 categories. There are many proto-oncogene. I am not repeating all. These all are proto-oncogene. You have to identify them in the classification. The 3 are important that we have covered today. So what are the 3 important proto-oncogenes we have covered today? The first one is the RAS. The second one is ABL-BCR. And the third one is MYC. These 3 I have given you the details. These 3 I have given you the details. In each of them, first I have told you what happens normally. What happens normally. And then I have told you what happens during mutation. And then I have told you the examples of them. Examples of them. Let me revise. Let me revise. In RAS, we have seen normally what happens. Normally, RAS is bound with GDP. So it is inactive. When growth factor is coming, at that time, RAS become active. GDP is replaced by GTP, and this active RAS will do one mitosis via various mechanisms in the cytosis. Immediately after one mitosis, the gap inside the RAS will be activated, and gap convert active RAS into inactive. GTPase will come in the role. But during mutation, gap is non-functional. Since gap is non-functional, so RAS become active permanently. That will lead to uncontrolled mitosis, that will lead to cancer. So that is normal and abnormal in RAS. We have seen the 3 examples of RAS. What are the 3 examples of RAS? KRAS, KRAS, NRAS, and HRAS. Learn the name of the cancers they are causing. KRAS causes pancreatic cancer and breast cancer. NRAS causes AML and neuroblastoma. HRAS causes bladder and kidney cancer. So learn the name of the organs. So that is the RAS. That is the RAS. Coming on ABL-BCR. In ABL-BCR, we have already seen that normally ABL is present on chromosome 9 and BCR is present on chromosome 22. Let me cover this also. Here MYC is present on chromosome 8 and immunoglobulin is present on chromosome 14. But during mutation here is translocation. Here the mutation was point mutation. In the other 2, the mutation is translocation. So here in translocation, ABL will come to BCR on 22. Here MYC will come to immunoglobulin. So here ABL BCR hybrid is formed on 22. Here MYC immunoglobulin hybrid is formed on 14. So here chromosome 22 is known as Philadelphia chromosome. Here no special name is given to 14, but here Philadelphia chromosome. Give me a thumbs up. So that leads to uncontrolled mitosis. This combination leads to uncontrolled mitosis and that leads to cancer. Now let me tell the examples. The examples are very easy. If I talk about ABL BCR, it is only one CML. No other types, only one type CML. The treatment is imitinib. Treatment पे भी question आता है. You have to learn the targeted therapy imitinib. Let me come on the examples of MYC. The MYC is also three type like RAS. So it is CMYC, NMYC, LMYC. L for lung, small cell carcinoma of the lung. N for neuroblastoma and C for Burkitt. Burkitt. In तीनों पे question है. Everyone give me a thumbs up. No one in this world will explain you all the proto-oncogenes with such a super simplicity. So should I proceed ahead? I am done with proto-oncogenes. I would like to learn some questions from proto-oncogenes. That is the accelerators. Are you people ready for the questions? This is the first question in front of you. This is the first question in front of you. From the classification itself, you have to tell me the best example of proto-oncogene which is activated by point mutation. Now, all of them are proto-oncogenes. But tell me the proto-oncogene in which translocation. Now, there are various types of mutation. Now, I have told you three types of mutation. Point mutation, translocation and amplification. So, which among all of them are proto-oncogene? I am not asking which is the proto-oncogene. So, RAS, NMYC, LMYC and ABL. But I have told you ABL BCR में translocation होता है. MYC में भी translocation होता है. But in RAS, it is the point mutation. It is the point mutation of gap. So, yes, Z. The correct answer here is A. So the correct answer here is A. It is the previous year question from AIMS. Right. Coming to the next question. You will tell me what happens in Philadelphia chromosome. What is the Philadelphia chromosome? Chromosome number 22. Chromosome number 22. So what is happening in that? Is it balanced translocation taking it? So how Philadelphia chromosome is formed? Is it due to balanced translocation? Is it due to deletion? Is it due to non-disjunction? Or is it due to duplication? Why it is formed? You tell me why Philadelphia chromosome is formed. Everyone can participate in the polls. Come on. At max, you will be wrong, but please participate. So, yes, tell me what is happening in Philadelphia chromosome. Yes, Z. Again, the answer is A. It is the balanced translocation between 9 and 22. On 9, we have ABL and on 22, we have BCR. So, there is balanced translocation. Some portion of 9 is given to 22. In exchange of it, some portion of 22 is given to 9. So, yes, the correct answer here is A. So, it is leading to CML. Correct answer here is A. Yes, Manoj. Z, both of you are right. The correct answer here is A. Very good. So this is the next question in front of you. Amplification of NMYC leads to. I have told you there are three types of MYC. CMYC, NMYC, LMYC. I have told you the cancers by each of them. I have told you the cancers. So please tell me what is the correct answer here. I am asking about NMYC, the N one. So NMYC leads to neuroblastoma, retinoblastoma, osteosarcoma or neuroma. Yes, what does it lead to? Yes, what does it lead to? Again, the answer is A. Very good, very good. So N for N, it is neuroblastoma. L for L, it is lung. But C is for B, it is Burkitt. It is Burkitt. Have you got it? So correct answer here is A. The correct answer here is also A. Very good. Both of you absolutely right. Right. So C will lead to Burkitt. L will lead to small cell carcinoma of the lung. L for L. And N leads to neuroblastoma, right? So C for B, I learned like this, C, L, N. L for L, N for N, but C is for B, B for Burkitt. So learn like this, you will never forget. So this is the next question based on the classification of proto-oncogene. Please tell me, what is the correct answer here? Yes. So all are growth promoting proto-oncogenes. All are growth promoting genes. Growth promoting genes means accelerator. All of the following are accelerator except. So which of the following is not a proto-oncogene? I guess this question you will not be able to do if you don't know the classification properly. Yes. So in the classification, what was not there? I'm asking to you. Yes, Z, you are right. You are right. So in the classification, TNF beta is not there. Rest all the things. TNF alpha is there, PDGF is there, and FGF is there. I will show you the classification. See the classification. In this classification, if you mind, it is TNF alpha. It is not TNF beta. Let me clarify you. TNF alpha is a proto-oncogene. These all are proto-oncogene. But TNF nahi TGF. TGF alpha is a proto-oncogene, but TGF beta is the tumor suppressor gene. So here TGF beta is a tumor suppressor gene, but TGF alpha, can you see FGF? And what is the third option? PDGF. And this one is the PDGF. These all are examples of proto-oncogene, but not TGF beta. So classification is very important. So next question in front of you, what is MYC gene? I know it is a proto-oncogene, but which category of proto-oncogene? There are various categories. Now it is acting by via which mechanism? So is it a growth factor? It is a growth receptor. Is it a signal transduction? What is it? It is transcription. What is it? How does MYC gene act? What is the activity? See in the classification where it is coming. Here it is coming. It is coming in transcription protein. It is one of the transcription protein. So the answer here is D. It is the transcription protein. In the classification, yes, very good Z. So the correct answer here is D. Yes, Manoj. The correct answer here is D. Have you got it? Have you got it? So this is the thing. Which of the following is a tumor promoting effect? Tumor promoting effect means the accelerator. So which of the following is a proto-oncogene? First, understand the meaning of the question. Which of the following is a proto-oncogene? Read the four options are in front of you. Is it BRCA? Is it retinoblastoma? Is it MYC or is it P16? So you have read the classification of proto-oncogene. You tell me which of the following is coming in the classification of the proto-oncogene. Which of the following is coming in the classification of proto-oncogene? I am waiting for your answer. What is the correct answer here? Yes, of course it is MYC. The MYC is coming in the classification. Rest 3 are tumor suppressor genes. Yes, Z. The correct answer here is C. Rest 3 are tumor suppressor genes. So you can see MYC, the 3 type of the MYC are tumor proto-oncogene. Rest all are tumor suppressor gene. No, the answer is not A, Manoj. The answer is C. BRCA gene is a tumor suppressor gene. You got it? So coming to the next question. The next question is in front of you. I want to see who is answering this correctly. Tell me, in RAS, the MAPK pathway in RAS is counteracted by. Tell me the mechanism, how RAS become inactive. MAPK pathway is the activation of RAS. How does counteracted? Counteracted means how RAS become inactive. RAS को inactive कौन करता है? Normally, who does the inactivation of RAS? Read the four options. Is it protein kinase C? Is it GTPase activating protein? Is it phosphatidyl inositol or is it inositol triphosphate? Who is causing? Yes, B. It is GAP. GAP is GTPase activating protein. Again, it is a difficult level question from previous year papers of the AIIMS. So it is a difficult level. So if you don't know the detailed mechanism, you can't answer this question. Give me a thumbs up. So correct answer here is B. Correct answer here is B. Come to the next question. Now read the question. You tell me the answer. What is false about proto-oncogene? Which statement is false? Read the four options. Which statement is false about proto-oncogene? Who will tell me which statement is false about proto-oncogene? Tell me the false statement about proto-oncogene. Tell me the false statement. What is the false statement? You tell me. So I'm asking about proto-oncogene. Write your answer in the chat box. Is it important for normal growth? Yes, proto-oncogenes are required for normal growth. May be converted into oncogenes once they are mutated. Yes, they are converted into proto-oncogene got converted into oncogene once they are mutated. Right? c-MYC overexpression leads to lymphoma. Yes, C for Burkitt lymphoma. Burkitt lymphoma is a type of lymphoma. So no Z, it is also right. Their mutation causes retinoblastoma. No, retinoblastoma is due to mutation of RB gene and it is not a proto-oncogene, it is a tumor suppressor gene. That's why incorrect among them is D. D is incorrect. It is not C which is incorrect. Why you all are saying C? It is D. I don't know why you have said C. Both of you are saying C. C is not the answer. D is the answer. C is correct now. c-MYC is Burkitt lymphoma. Burkitt lymphoma is one of the lymphoma now. So c-MYC is causing Burkitt lymphoma. Right or wrong? Have you realized your mistake? Z and Manoj, both of you. Have you realized your mistake? The correct answer is C. Correct answer is D, not D. Answer is D. Because D is incorrect. False is asked now. Give me a thumbs up if you got it. Proto-oncogene do not lead to retinoblastoma. Tumor suppressor gene is leading to retinoblastoma. That's why incorrect is D. Question is about proto-oncogene. Have you got it? So correct answer is D. This is the next question in front of you. Following are required for normal growth except. For normal growth, what is not required? For normal growth, what is not required? I have told you for normal growth, we have 4 types of gene in human body. So we have accelerators, that is proto-oncogene. We have brakes, that is tumor suppressor gene. We have mechanics, that is DNA repair gene. And we have the destroying factory. The destroying factory that is apoptosis regulating gene. These 4 are required for normal growth. Which is not required for normal growth? Proto-oncogene are required. Tumor suppressor genes are required. These are the accelerators. These are the brakes. DNA repair genes, these are the mechanics. But oncogenes are required for cancer, not for normal growth. When proto-oncogene become overstimulated, it leads to oncogene. So oncogene is it is seen in Cancer, not normal. Here we are asking for normal. So yes, both of you are right. The correct answer here is C. Have you got it? Have you got it? So I am done with proto-oncogene. I am done with proto-oncogene. All the important points of the proto-oncogene are done. Give me a thumbs up. Now I am starting with tumor suppressor gene. Now I am starting with tumor suppressor genes. What are tumor suppressor genes? As I have already told you, these are the brakes. These are the brakes. So these are the brakes for cellular proliferation, right? So in them, due to mutation, loss of function mutation takes place, and they become inactivated. That they leads to cancer. So you can see these are the tumor suppressor genes. These are the brakes. Normally they inhibit mitosis, but once they become mutated, they are inactive. Now they are not inhibiting mitosis, so it will lead to uncontrolled mitosis. It will lead to cancer, right? So we already know that, right? Now classification. It is also divided into 5 categories like proto-oncogene. This is the classification of tumor suppressor gene. What is the classification? The first is the retinoblastoma gene. C. It is a tumor suppressor gene. The second is P53. It is also a tumor suppressor gene. The third is TGF beta. If you have noticed, TGF alpha is a proto-oncogene. TGF beta is a tumor suppressor gene. The fourth is the APC and beta catenin and others. The fifth is the others. Others में क्या क्या आता है? BRCA1, BRCA2, very important. WT1, WT2, Wilms tumor 1, Wilms tumor 2, neurofibromatosis 1 and 2, and VHL, Von Lennedue Hippel, Von Hippel Lennedue. So please now. So these are, that is the classification. Now compare the classification of proto-oncogene and tumor suppressor gene. You have to learn the classification. There is no escape. There is no escape. It is also divided into 1, 2, 3, 4 and 5 categories. It is also divided into 1, 2, 3, 4 and 5 categories. Let me try to remember it. Let me try. It is difficult, but I will try for you. So proto-oncogene, we have growth factors, we have growth receptors, we have signal transduction proteins in the cytoplasm, we have transcription, nuclear transcription proteins, and we have cell cycle regulators, right or not? Here now learn the name of the genes. I am not saying the name of the genes inside them. There are many genes. Growth factor में 4 genes थे, PDGF, TNF alpha, TGF alpha, EGF, and one more was there, FMS, I guess. One more was there. So you have to learn the name of the genes. Signal transduction में 2 ही थे, which I have explained you. RAS I have explained you, and ABL BCR I have already explained you. The nuclear transcription में एक ही था, MYC, the 3 type of the MYC. So in this way, you have to learn the classification. Tumor suppressor gene is also divided into 5 categories. The first is the retinoblastoma, very important. The second is the P53, very important. These two I am going to teach you in detail. Here I have taught you 3, here I am going to teach you 2. That is the second one, P53. The third one is the TGF beta. यहाँ पे TGF alpha था, यहाँ पे TGF beta है. The fourth is APC and fifth में others. Others में BRCA1, BRCA2, WT1, WT2, BRCA1, BRCA2, WT1, WT2, that is Wilms tumor 1, 2. This is breast carcinoma 1, 2, and neurofibromatosis NF1 and 2. These are the important and others. Give me a thumbs up. I tried hard. You have to learn the classification. There is no escape. You have to learn. Now in proto-oncogene, I have given you the details of RAS, ABL, BCR and MYC. Here, I will give you the details of retinoblastoma and P53. Let me clarify one more concept here. Let me clarify. Among proto-oncogene, the most common gene which is mutated is RAS. The answer is RAS. Among tumor suppressor gene, the most common gene to be mutated is P53. And overall, among all genes, the most common gene to be mutated is again P53. So there are 3 questions. The most common proto-oncogene to be mutated, the most common tumor suppressor gene to be mutated, and the most common overall gene to be mutated. So 3 questions, 3 answers. 3 questions, MCQ questions with 3 answers. Who will tell me the 3 answers? Proto-oncogene, it is RAS. The most common proto-oncogene to be mutated is RAS. The most common tumor suppressor gene to be mutated is P53. It is a tumor suppressor gene. And overall also it is P53. Give me a thumbs up. Everyone give me a thumbs up. Okay. Now I will teach you 2 genes here. The first is the retinoblastoma. Next is P53. One by one we will draw the flowcharts of them. Now retinoblastoma is a difficult level gene. Right. Again I will try my best. Let me see how I can teach you retinoblastoma. Let me start retinoblastoma gene. RB gene. The retinoblastoma gene. The RB gene. It is present on chromosome number 13. Now humans have, this is a human cell. This is the nucleus of human cell. As we all know, we have 23 pairs of chromosome. Currently, I am interested in pair number 13. We have 2 chromosome 13. A pair, 2 alleles of chromosome number 13. So on both of them, we have RB gene. So basically all humans have two RB gene. How many RB genes are you are having? You are having two. How many I having? I'm also having two. So all human beings have in each cell, we have two RB gene. Give me a thumbs up. In each cell, we have two RB genes. We have to learn the meaning of the two, right? We have two RB genes on both chromosome number 13, right? It is the first tumor suppressor genes to be discovered, right? That is the introduction. Okay, again the two scenarios. What happens normally, how it functions normally, and what happens during mutation. During mutation, how it leads to the cancer. So what happens during mutation? I will tell you the two scenarios one by one. Let me start normally. Let me start normally. How does it function normally? How does it function normally? Let me start. So listen normally what is happening. Listen normally what is happening. Now what is retinoblastoma gene? Retinoblastoma gene is a tumor suppressor gene. It is not a proto-oncogene. Please mind my words. Listen, Manoj Z, listen. It is a tumor suppressor gene. So it is a brake. What is its normal job? It is not accelerator, it is brake. Normal job is to decrease the mitosis. Normal job is to decrease cell division, right? Normally. So normally in the flowchart, I will show you how it is decreasing the cell division, right? The diagram is not given. I will draw the diagram for you. Okay. Okay. I will draw the diagram for you. Don't worry. So I will draw two cells for you. So this is the first cell. This is the nucleus of the first cell. This is the second cell. This is the nucleus of the second cell, right? In the first cell, growth factor is coming. Growth inhibitor. I will do growth factor or growth inhibitor. Okay, growth inhibitor is coming and binding here. In the second cell, growth factor is coming and binding here, right? Now in the nucleus, you can see this is chromosome number. I'm sorry, the diagram is gone. I have to draw it again. I'm sorry. I'm sorry. Give me a minute. Am I visible, audible? Can you see me? Just a minute. I will draw it again. Don't worry. Okay. So let me take a blank sheet again. Another blank sheet now. How it will go? ये कैसे जाएगा? Okay. Okay. Just a second. Okay. So here I am. So listen, I'm drawing the two. This is the nucleus or cell. First cell. This is second cell. In the second cell, this is the nucleus. You can see this is chromosome number 13 here, and this is chromosome number 13 here. On chromosome number 13, this is the retinoblastoma gene here. Right, this is the retinoblastoma gene, and here on chromosome 13, this is the retinoblastoma gene. Why I'm taking two scenarios? I'm teaching you normally. Normally, two things can happen. What two things can happen? Normally, growth inhibitor can come and growth factor can come. Either growth factor is coming or growth factor is absent or growth inhibitor is present. In which of the following scenario retinoblastoma gene will be activated? Retinoblastoma gene कब activate होगा? Please consider retinoblastoma gene is a tumor suppressor gene. So when it will be activated? When growth inhibitor is coming, it will be activated, or when growth factor is coming, it will be activated. My common sense question to you. Apply your common sense and tell me the answer. It will be activated since it is a tumor suppressor gene. It will be activated when growth inhibitor is coming. When growth inhibitor is coming. So when growth inhibitor is coming to this cell, retinoblastoma cell will undergo hypophosphorylation. It will bind with few phosphorus groups. Hypophosphorylated retinoblastoma gene is active form. Like for RAS, in RAS gene, when it is binding with GDP, it is inactive. When it is binding with GTP, it is active. I have told you, na, in the similar way, retinoblastoma gene, when it is hypophosphorylated, it is active. And when it is active in hypo, and when it is hyperphosphorylated, it become inactive. Give me a thumbs up. So when growth inhibitor is coming, it is hypophosphorylated. It binds with few phosphorus group. So that's why it become active. When it is active, what actually it is doing? See what it is doing. It will hold elongation factor 2. This is elongation factor 2. Can you see a factor? Okay, let me draw. This is elongation factor 2. This factor is required for mitosis, without which mitosis do not take place. So it is inhibiting elongation factor 2 from doing mitosis. So it is doing its job. It is active. It is inhibiting the cell growth. It is inhibiting the mitosis. Have you got it? What will happen when growth factor is coming? When growth factor is coming, can you see this is growth factor? When growth factor is coming, there is hyperphosphorylation of retinoblastoma gene. Everywhere it is bind with phosphorus, multiple phosphorus. It become hyperphosphorylated. Once it become hyper, now compare, it is hypophosphorylated, this is hyperphosphorylated. It is active form, it is inactive form. So it become inactive now. So once it become inactive, Once it become inactive, it will release elongation factor 2. It will not hold elongation factor 2 anymore. And elongation factor 2 on releasing, it will perform the mitosis. So mitosis will happen. Once it become inactivated, so when retinoblastoma is active, it is inhibiting mitosis. When retinoblastoma is inactive, it is causing mitosis. Give me a thumbs up. So can you see the two flow charts? Everyone give me a thumbs up. I tried hard. At least I tried. So when growth inhibitor is coming, there is hypophosphorylation of retinoblastoma gene. It become hypophosphorylation. See where is that retinoblastoma gene? It is like this hanger. Can you see this hanger? It is retinoblastoma gene. See how many phosphorus groups it is attached? Only two. Only two groups are attached. So that's why it is active. Since it is hypophosphorylated, it is active. So once it is active, it is holding E2F. This is E2F inside this. It is holding. उसको पकड़ के बैठा है, छोड़ ही नहीं रहा है. So the retinoblastoma gene is holding E2F and it is blocking the transcription because E2F is not free for doing the transcription. It is not free for doing the mitosis. Now see the other way around. When growth factor is coming, instead of growth inhibitor, there is growth factor. So instead of hypophosphorylation, there is hyperphosphorylated. There is hyperphosphorylated retinoblastoma. See, this is retinoblastoma gene. See, this is retinoblastoma gene. How many phosphorus groups it is attached? See, so here only two were there. Here there are multiple. So it is hyperphosphorylated. Hypo is active. Hyper form is inactive. It become inactive now. Once it will become inactive, it will free the E2F. उसको छोड़ देगा कि मैं तो inactive हूँ, जा जी ले अपनी जिंदगी, right? So this E2F become free now. Now once it becomes free, it will go and do the mitosis. Give me a thumbs up if you got it. So these two scenarios are taking place normally. Let me tell what happens in mutation. Let me tell you what happens. Give me a thumbs up if you got till now. Let me tell you what happens during mutation. In mutation, permanent hyperphosphorylation of retinoblastoma. Permanent hyperphosphorylation. So during mutation, permanent hyperphosphorylation of the gene. Now whether growth factor is coming or growth inhibitor is coming, it is hyperphosphorylated only. It remains hyperphosphorylated permanently. That is permanently it become inactive. Permanently it become inactive because it is hyperphosphorylated permanently. It is inactive permanently. So permanently E2F is free. Now E2F is free. It is dislocated permanently. So it will cause uncontrolled mitosis. This E2F will cause uncontrolled mitosis and that will lead to cancer. Give me a thumbs up. I really tried hard here. Come on. So when growth inhibitor is coming, there is hypophosphorylation. It become active. It holds E2F. It forms a complex with E2F. And since E2F is not free, it is inhibiting the mitosis. There is no mitosis. But when growth factor is coming, instead of hypo, it become hyperphosphorylated. Instead of active, it become inactive. So it dissociates from E2F. E2F will become free and this E2F will cause the mitosis. So this is normally. But during mutation, there is permanent hyperphosphorylation of retinoblastoma gene, which will lead to permanent inactivation. It will lead to permanent inactivation of retinoblastoma gene. Now, this E2F is dislocated permanently. This E2F is dislocated permanently and this dislocated E2F, it is free permanently. It will cause uncontrolled mitosis. It will lead to cancer. It will lead to cancer. I really tried hard. I want the thumbs up from you, right? So that is the thing. That is the thing. If you got this thing, I would like to tell you two more things in retinoblastoma. Two more terminologies in retinoblastoma gene I would like to tell you here before ending the lecture. I would like to tell you here. Have you got it? Have you got it till now? Okay. Let me continue now. So in retinoblastoma, there are two things: loss of heterozygosity and Knudsen two-hit hypothesis. What do you mean by these two terminologies? Loss of heterozygosity क्या होता है? Knudsen two-hit hypothesis क्या होता है? Okay. Let me first teach you Knudsen. Knudsen is the name of the scientist who has given a hypothesis. That's it. That is Knudsen two-hit hypothesis. As I have told you, we have two chromosome 13. On two chromosome 13, each of which have one retinoblastoma gene. So we have two retinoblastoma gene, one on each allele. Two retinoblastoma alleles, one on each chromosome, right? So when one of them is mutated, will the person have cancer? No, the person will not have cancer. When both of them are mutated, then the person will have cancer. So that is the theory given by the Knudsen. Knudsen two-hit hypothesis. So Knudsen is saying when both the retinoblastoma genes are mutated, then only the person will have cancer. Which cancer? Retinoblastoma cancer of the eye. Cancer of the eye, that is retinoblastoma cancer. So when both the normal alleles of retinoblastoma are mutated, then only the person will have retinoblastoma. So that is two hit. The first hit, the second hit. Hit means mutation. Hit means mutation. Give me a thumbs up if you got it. Right. Have you got it? Have you got it? Now there are two types of retinoblastoma. There are two types of retinoblastoma. What are the two types of retinoblastoma? One is sporadic, one is familial. One is sporadic retinoblastoma and one is familial retinoblastoma. These are the two types of retinoblastoma. Okay, let me explain you the both one by one. It is very easy to understand. Listen, this is a mother. She is a girl having 2 chromosome 13. And this is a father having 2 chromosome 13. From the mother, ova is formed. And from the father, sperm is formed. So fertilization takes place and zygote is formed. Can you see zygote? So zygote is getting 1 chromosome 13 from the mother and 1 from the father. So all embryos, zygotes, fetus have 2 chromosome 13. On each chromosome 13, one retinoblastoma gene. So one retinoblastoma gene coming from the mother, one from the father. Both are yellow in color. Yellow means normal. So mother is also normal, father is also normal. They got married. So sperms and ova are also normal. And because of the fusion, the fetus formed is also normal. So at the birth, the fetus is absolutely normal. The child is absolutely normal at birth. After birth, one of the gene got mutated. In retinoblastoma gene, one of the gene got mutated, other is still normal. The other is still normal. So this is a carrier. The child is known as a carrier. But child will not have the cancer. The child is a carrier. So this is the first hit, first mutation. And after that, when second hit or second mutation takes place, that is both the retinoblastoma genes are mutated, then in the eye, the child will have a cancer, that is retinoblastoma. So here, this is the sporadic form. Both the hits are after birth. The first hit, the second hit. Both the mutations are after birth, that is known as sporadic. Give me a thumbs up, that is a sporadic one. Now come on the familial form. In the familial form, what is happening? In the familial form, we have, you can see a father. The father is, you can see the mother. The mother, both the genes are yellow. Can you see? Both are yellow. So the mother is normal. So the ova formed is normal. This is the ova, it is normal. But see the father. Now please see the father. Inside the father, one of the gene is already mutated. The father is a carrier. But how does the father know that I am a carrier? No one do the genetic testing before marriage, na? So he's a carrier. He don't have cancer. He don't have cancer. So this is a boy who is a carrier. He got married with a normal girl. So the ova is normal. But can you see the sperm? Inside the sperm, it is the mutated retinoblastoma gene. So in the zygote, one is mutated coming from one of the parent and one is normal. So at birth, the child is a carrier. The child is a carrier. See the previously, at birth the child was normal there. Here the child is a carrier at birth. Child don't have cancer, but here the child is a carrier. And after birth, only one mutation is required to cause the cancer. So in familial form, first hit takes place before birth. It is coming from one of the parent. And second hit is taking place after birth. And in sporadic form, both the hit, the first and the second, takes place after the birth. So that is the difference between the two types. Have you got it? So sporadic form and familial form. Familial form. These are the two types of retinoblastoma. In sporadic, both the mutation, the first and second, occurs after birth. After birth. In familial, the first is coming from one of the parent before birth and second is happening after birth. Second. Now, does it make any difference? Now, just suppose you are a pediatrician or you are a pediatrician or you are an ophthalmologist, the parents are carrying their child to you. This is the child, parents are carrying their child to you that doctor, our child is having some tumor in one of the eye. You are doing the examination, you are doing the investigation, it is coming out to be retinoblastoma. Does it matter for you, for the parents, for the child, whether it is sporadic or whether it is familial? Does it matter? How does it matter? If it matters, how does it matter? Now the child is blind by one of the eye. Retinoblastoma is a malignant tumor. You have to remove entire eye. So after surgery, the child is going to be blind with that eye. Now, if it is familial, it is going to be bilateral. That is the bad news for the parents. It is the bilateral, it is going to happen in other eye in future. But if it is sporadic, it is going to be unilateral only. It is going. So, एक आंख तो गई, लेकिन दूसरी जाएगी कि नहीं जाएगी, that depends whether it is sporadic or familial. How you will come to know whether retinoblastoma is sporadic or familial? In this child, whether retinoblastoma is sporadic or familial, you cannot say that by looking at the child. You have to test the parents, the mother and the father. You have to test the parents. The parents, if both the parents are normal, it is sporadic. And if one of them is carrier, it is familial. So you can do that. That is the first difference. The second difference, you can see familial and sporadic. Have you got it? In familial, one mutation occurs before birth from a carrier parent and other mutation occurs after birth. But in sporadic, both the mutations occurs after birth. Right? That is the first difference. Familial constitute only 40%, sporadic constitutes 60%. Familial is bilateral and sporadic is unilateral, right? So familial is bilateral, sporadic is unilateral. The last difference, the familial one, apart from retinoblastoma, there is other cancer risk is also there, other organ cancer risk. So this child having 200 times more risk of developing osteosarcoma, and there is no other cancer in sporadic one. So that is the difference. Have you got it? Have you got it? So that is the thing. That is the thing. We are done with retinoblastoma gene also. So here I would like to stop. We have covered the genetic regulators. We have covered the genetic regulators of neoplasia, the molecular genetic mechanism of neoplasia. Thank you very much for being with me. I really enjoyed teaching you. Hopefully you have learned in today's class. That is the various genes. Don't forget to write your feedback in the comment box. Thank you very much. My next class will be there tomorrow, in which I'm going to continue the series. Thank you very much. If you like the lecture, don't forget to click on the like button before you leave the lecture. Please share the links with your friends, colleagues. Subscribe our YouTube channel, Let's Crack NEET PG, and don't forget to press the bell icon for the notifications. Right? Unacademy is offering. Okay, what you are asking, Z is asking, ma'am, why higher number of cases are sporadic? No, it is incidental. There is no reason for this. Z, there is no reason for it. It is incidental. Even I don't know the reason why the cases are more, but yeah, the prevalence wise, we have seen that. There is no reason for that, right? Particularly. So on Unacademy, there are few new offerings for the students the team is offering to you. If you take the subscription, you know on the app, previous year questions are available. You can do the practice of NEET PG, FMG, and INICET practice papers. Exclusive doubts clarification batch is coming next month. So you can from 6th April onwards. So you can ask your all the doubts to your educators, and they will clarify all your doubts, right? Any subject doubt. And after every plus class, we will give you a bunch of 5 to 10 questions to do the practice, right? That is the thing. And the biggest announcement is that only few hours, only 3 hours are remaining. So till 12 o'clock today night, 31st March, aaj raat ko 12 baje, this offer is valid. So if you take the subscription in the next 3 hours, you will get 20% discount instead of 10% discount. From tomorrow onwards, again, you will get only 10% discount. So if you want to plan, if you are planning to take the subscription in the next few days, take it today only in the next 3 hours. If you apply my code Sachdev10 before payment, you will get straightforward 20% discount instead of 10. So the code is SACHDEV, Sachdev10. These are the various plans available with the Plus and Iconic. You can see the price. So thank you very much. In Plus subscription, Unacademy gives you only live and recorded lectures of Unacademy. In Iconic, along with Unacademy, we will give you access of Prep Reader also. Once you take the subscription, you will be eligible for all these batches. And thank you very much for being